There we go. Yes. Our immune system is doing a remarkable job in freeing us from infecting viruses and bacteria. And this happens through a double process, one immediate part which involves inflammation and which is not very specific, followed by an extremely specific phase involving B lymphocytes and antibodies, and T lymphocytes which kill the infected cells. The regulation of the whole process is, in my view, rather clumsy, involving tens of activating or inhibitory receptors and their ligands. But it works, and therefore it's not a surprise that since the beginning of the 20th century, one has considered the possibility to try to utilize the immune system to free us from cancers. The crucial question in that respect was whether cancer cells would bear the so-called tumor-specific antigens, that is, highly specific antigens that would not be present on normal cells and could be recognized by T cells which kill the cells they recognize. And that question was for a long time undecided, but now has been decided in a positive way about 30 years ago. And to tell you this, I should ask you to look at the first slide, which explains more or less how T cells recognize their target. Imagine that you have a viral genome shown in the nucleus there, which will produce the mRNA, the protein. The protein will be often degraded by the proteasome, and small peptides will move to the endoplasmic reticulum, where they will bind to class 1 HLA molecule, which will travel to the surface to form the following pattern with the small peptides. You see here in gray the HLA molecule and the small peptide, which each amino acid represented in the middle. And this is what the T cell receptor is going to recognize. Now, what became clear 30 years ago was that this happens not only with a viral genome, the products of a viral genome that has infected us, but also with the normal products of the cell. In other terms, the T cells exert a real immunosurveillance of the integrity of our genome. If you have a mutation in the genome, it will make an abnormal protein, an abnormal peptide, and then this new sequence will be recognized by T cells which have not been eliminated by natural tolerance, which eliminate the T cells that recognize our normal component. Well, it turns out that many cancer cells are full of genetic mutations, and that these are important sources of new antigens, and these antigens are totally specific for the tumors and, by and large, for individual tumors. Now, there is a second mechanism that produces tumor-specific antigens, and that is the so-called cancer germline genes. There is a set of genes that is expressed only in tumors and in early germ cells, spermatogonia, for instance. So there you can also get new peptides against which we are not tolerant. Why are they completely cancer tumor specific? Because the early germ cells that express them do not carry HLA class 1 molecule. So they have the proteins, but they do not expose the peptide on their surface. Then, of course, a third, but rather obvious type of tumor-specific antigens is the antigens that you find on cancer cells that are the result of a viral transformation. Now, when that was established, the logical next step was to try to use vaccination. And I hope I was, yeah. That was started in the beginning of the 90s, And vaccination is, of course, the absolutely ideal immunotherapy because it's entirely specific and therefore very largely devoid of harmful side effects. And this is exactly what happens, what we saw in initial patients. In those few patients who responded, there were absolutely no harmful side effects. But vaccination is not easy to apply because you have to identify the antigen on the patient, on the tumor you're trying to treat. That is a little easier with the cancer germline genes, which are common to a number of different tumors. But the main drawback of vaccination was that, for instance, in metastatic melanoma, it worked only on a very small proportion of patients, something like 8%. And this was clearly not enough for it to take a foothold in usual medicine. The reason why it doesn't work better is, in my view, shown on the next slide. It turns out, and it took us a long time to realize this, that many patients we were trying to vaccinate had already made a spontaneous anti-tumor T-cell response. But this response gets stuck. It stalls, and the tumor then has these inactive lymphocytes combined with a strong immunosuppressive environment. And most probably, the lymphocytes we try to introduce into the tumor as a result of vaccination get caught in this immunosuppressive environment, and they get silenced. Fortunately, 10 years ago, Jim Allison and, sorry, this is not the right slide. Jim Allison and Takashi Hondo developed a new idea, which was to use a set of inhibitory receptors, namely CTLA4 and PD-L1 and PD-1, and try to inhibit these inhibitory receptors, which would activate the T lymphocytes. And this has given extremely interesting results. In theory, it's rather convenient to apply because you can apply the same inhibitory antibodies to all the patients. Second, unfortunately, it's not the dream immunotherapy because you're activating all sorts of T cells, not just those that are specifically directed at the tumor-specific antigens. And as could be expected, this therapy is accompanied by often by autoimmune effect that can be quite harmful, but that appear to be manageable. But most of all, this therapy was effective in a much larger proportion of the melanoma patients, for instance, as was said a few minutes ago, and therefore took hold into the practical cancer treatment. And I think that this is very important because now that these results exist, the activity in trying to improve immunotherapy is going to be very considerable. I believe that from now on, we are going to have steady progress, and that one very fruitful way is to try to be clumsy like the immune system. That is, not to try to relate on mono-immunotherapy, but try to add agents, especially agents that will remove the local immunosuppression, that will counteract the local immunosuppression. And I'm quite optimistic that in the years ahead, we are going to see very considerable progress. A curious point I want to make is the following. When you look at how incredibly difficult it should be for the immune system to get us rid of bacteria and viruses, which multiply very rapidly and have all sorts of tricks to avoid the immune attack, you're really amazed that it works, that we get cured, that we heal from infections. But the reason is, of course, that there has been a fantastic amount of natural selection in favor of developing the best process. When you look at how easy it should be to eliminate cancer for the immune system, to eliminate cancer cells that grow very slowly compared to bacteria and viruses, and that have, after all, proper antigens, you feel that it should be a child's play for the immune system to do it. And it doesn't do it, and there is only one good explanation for that, and that in the history of mankind, there hasn't been the slightest selective pressure instead in favor of trying to get rid of cancer. Cancer affects mostly people in old age after their reproductive period, and so there is absolutely no interest in nature in getting us rid of cancer, unpleasant as it is. I would not even totally exclude that there has been a counter-selective pressure that maybe it was good for human communities not to have too many old people around. Now, in the last, I see that I still have 3 minutes, and having said what I think I should tell you scientifically, since the meeting comprises thoughts about the transformative aspect of science on society, well, clearly, if one is optimistic and this leads to enormous improvement in cancer treatment, the permanent effect will be an increase in the proportion of older people in the population. Insofar as they keep good mental health, I think this is not a problem at all, and personally, I'm not at all convinced by the lamenting of economists who tell us that if we have too few young people to work for the old people, we're going to have severe problems. I think these problems are perfectly manageable, especially with the scientific progress we know. The transient effects are, the first one is the ruining of social security because of the cost of these treatments. There would be a lot to say about it, but I don't think it's now the time to say it. Let me only say that I think it's important to think about the transformative effect on science, on society, but I think that at the present time, there are quite strong transformative effects of society on science. For instance, in cancer, in Europe, incredibly stupid and demanding regulations by the European communities have almost completely stopped academic clinical research. Industries can do it, but the cost and the delays have been so much increased that academic research is strongly inhibited. And this is clearly an example where there is too much political interference, not understanding situation properly, and this will cost, in my view, thousands of lives. Another transient effect, of course, is the increase in the total population if many more people survive. And here I will venture one last remark, which is not particularly related to my subject, but I take the 1.5 minutes that's left. I believe that high increase in human populations are manageable. But what I believe is a road to disaster is the accumulation of this excessive population in mega cities. I think that's a recipe for disaster and for human unhappiness and for anti-ecological behavior. And so Dr. Schnellhuber said we should not only complain about the bad things that happen if we don't act properly, but we should propose positive things. I think one of the most positive things we could propose, and I think where science has an enormous role to play, is to reinvent new, smaller cities where people can move. I can tell you that in France, there is lots of space in Auvergne and all that, where we could have marvelous cities of 50,000 people, and where people could keep their contact with nature, and where you would not have what I feel one of the main tendencies of megacities, which is to dramatically increase the gap in quality of life between the richest people and the poor people. If you live in smaller aggregates where everybody has an access to nature, even modest people can have a perfectly decent and dignified life. Excuse me for leaving my subject, but I think this is an important thing I think we should think about. Thank you.